So we've covered anatomy in the two prokaryotic domains. Now let's move on to eukaryota, which includes your cells as well as fungal and protist plant cells. I want to make a note, this video is sort of optional depending on how well you remember this information from your prerequisites. I know different students took their prerequisites a different number of terms or years ago. I'm basically in this video just going to go through all of these organelles. So if you remember all those organelles and you just need a refresher for your textbook, feel free to skip past this one. If your prereq material was longer ago and you don't remember what any of this stuff does, you probably should listen to this video. So brief outline, we're going to talk about the anatomy of eukaryotic cells. Then we'll talk about the different kinds of eukaryotic microbes. We'll talk about some aspects of their physiology, some medical considerations, and then we'll cover the evolution of eukaryotes. There are tons of organelles, but like I said, most should be reviewed from prereqs. So this is the part of the course that will be a reprieve for you if you have retained that information from your prereqs if it wasn't that long ago. So I'm going to breeze through these, and I'll mostly let you review the material using the textbook as supplement. The nucleus contains and protects all a eukaryotic cell's genetic information, in other words, the DNA. So the DNA has this protective envelope around it called the nuclear envelope that prokaryotic cells don't have. And that's really nice because we want to prevent damage to the DNA because that's what gives the instructions for building and repairing and functioning of the cell. So this extra protection protects that DNA from environmental onslaughts like UV light or chemicals in the environment that might be mutagens or something like that. The endoplasmic reticulum surrounds the nucleus. There is rough and smooth endoplasmic reticulum. The rough endoplasmic reticulum looks rough because it is studded with ribosomes. So remember, ribosomes read genetic material and translates that genetic material into protein. So the genetic material is a blueprint for protein. So the rough ER, or RER, is a site of protein production. The smooth endoplasmic reticulum, or SER, is where lipids are produced. Lipids can include phospholipids for the cell membrane, cholesterols. Cholesterol is a precursor for steroids and steroid hormones, and cholesterol is also important for regulating the fluidity versus stability of the cell membrane. Okay, so cholesterols are lipids, steroids are lipids, and steroids are, they're hormones. So some of our hormones are lipid hormones. So estrogen, progesterone, testosterone, cortisol, those are all steroid hormones. So those are all produced in the smooth endoplasmic reticulum. So this structuring, this ordering inside the cell makes sense, right? Because you have the nucleus in the middle, and that's where the genetic information is. You want the rough ER to be just outside of that because these ribosomes have to read the genetic material in order to pump out proteins. Okay? And then the lipids are produced here. So basically all surrounding the nucleus this is where all of these really essential molecules are synthesized. And then just outside of the endoplasmic reticulum is the Golgi apparatus. The Golgi apparatus takes material that was produced in the endoplasmic reticulum, packages it, and sends it off to wherever it is destined to function in the cell. You can think of it as the cell's post office in that way. So there's the genetic material that directs synthesis of protein synthesis just around the genetic material, lipid synthesis just outside of that, and then the Golgi apparatus takes all the stuff from the ER that was just built there, the lipids, the proteins, packages them, and sends them off to their final destination in the cell. Mitochondria are the energy production centers of eukaryotic cells. They use oxygen to help harvest energy from food, producing massive amounts of ATP for the cell. Now we'll get a lot into ATP next week when we cover metabolism. So just very briefly, you'll have to remember that ATP is the energy currency of the cell. So there's all these different forms of energy out there. The cell can't actually use all of them, but it can convert all of them to ATP, and the ATP is the form of energy that the cell can actually use to do all the things it needs to do: grow, repair, communicate, move, all of those things that require energy. It uses ATP for. Okay, so ATP, that energy currency of the cell, is produced in the mitochondria. So I said mitochondria are the energy production centers, but that actually is not accurate because we don't produce energy. So one of the laws of thermodynamics is that energy is never created or destroyed. It's always constant in the universe. It's just converted from one form to another. So what the mitochondria do is they take all of these different energy sources coming in, like there's many different kinds of fats, many different kinds of carbohydrates and sugars, many different kinds of proteins. It takes all of these different styles of energy, extracts the energy from all of those energy molecules, and converts it all to the same form, ATP. So that energy conversion for the cell happens in the mitochondria. Now remember, the rest of the cell can only use ATP. So when ATP production stops, it doesn't matter how much sugar is there, the cell has no access to any of its energy in the environment anymore, right? So it really needs this energy conversion center called the mitochondria. The mitochondria have two membranes. The outer membrane is just, simple membrane. The inner membrane has these invaginations and this waviness to increase surface area. All right, so there's a couple other organelles in this picture that I want to cover. I want to focus on, let's see, okay, peroxisomes. Here's a peroxisome in blue, and lysosomes. Here's a lysosome in green. They look the same, right, under a microscope. They don't actually have these colors. So peroxisomes and lysosomes look exactly the same, but they're very different in their function. So lysosomes are these little tiny bubbles or vesicles that are filled with digestive enzymes and acids. Those are the digestion centers for the cell. So the cell might bring in this giant food material and it has to break it down into simpler pieces, just like you do in your stomach. So that's what the lysosome is for. It's digestion of large food molecules into small food molecules. And then those small food molecules might go to, for example, the mitochondria to extract energy and convert the energy to ATP. And then peroxisomes, those are right here. They look the same, but their job is to mitigate the damaging effects of oxygen. So yes, you need oxygen to live, but what you may not have realized is that oxygen is actually toxic to cells. It creates free radicals in the presence of water, and it wreaks all kinds of havoc on a cell. And so the job of the peroxisome is to go around neutralizing all those toxic forms of oxygen that arise inside the cell. All right, so that completes the features that all eukaryotic cells have, but that prokaryotic cells do not. There are also some features that all cells have, both prokaryotes and eukaryotes, and we've already covered them, but I'll just point out here that all eukaryotic cells also have ribosomes. So you can see those, we talked about the ribosomes in the endoplasmic reticulum. There's also some ribosomes just floating around in the cytoplasm. So remember, prokaryotes had ribosomes too. These are the protein building factories. They read genetic material and they produce protein from the genetic blueprint. Now, prokaryotes also have ribosomes, but there are some differences. Prokaryotes have different and much smaller ribosomes than eukaryotes. Both eukaryotes and prokaryotes have DNA as genetic material. So all cells use DNA as genetic material. Both prokaryotes and eukaryotes have a cytoplasm, which is just the liquid that everything's floating in inside the cell. Both prokaryotes and eukaryotes have a cell membrane, right? A lipid bilayer. And both prokaryotes and eukaryotes have a cytoskeleton made of protein that retains its structural, the cytoskeleton is its structural support. Right? All right, so that encompasses what all eukaryotic cells have. And some of those things prokaryotes also have, and some of them they don't. Okay. So when you're studying for the anatomy quiz, I would recommend making a list. What are the things that both prokaryotes and eukaryotes have? What are the things that only prokaryotes have? What are the things that only eukaryotes have? Now let's cover a few more anatomical features that are unique to eukaryotes, but that only some eukaryotes have. So from here forward, we're talking about some things that are unique to eukaryotes, but not all eukaryotes carry these things. All right, so some plant and plant-like protist cells have additional cell structures, like chloroplasts, water vacuoles, and a cell wall. So chloroplasts, water vacuole, and cell wall, animal cells don't have them. So this is going to be plants, algae, and plant-like protists that have these. Many protists and all algae and plants can photosynthesize, which means that they have chloroplasts which harness the sun's energy and directs it to the production of sugars for the plants. Chloroplasts are sort of similar to mitochondria in a way. They have two membranes. They have this outer membrane that's really simple, and then an inner membrane that has a lot of surface area and makes all these complicated structures. Okay. Vacuoles can store and release water to help maintain a constant internal water pressure. They are also storage they're also storage for some nutrients. So the vacuole, this is a protist here. And then here is a contractile vacuole. This vacuole can expand and store water for later. And then if the cell starts drying out, it can contract and squeeze out its water into the cytoplasm so the cytoplasm retains a constant osmotic pressure. Some eukaryotes have a cell wall, which is functionally similar to prokaryotic cell walls, but is structurally different. So the plant cell wall, so plants and fungi have the cell wall. So these cell walls are not made out of peptidoglycan, and they're not made out of pseudomurin. They're not even made out of a disaccharide, but rather a polysaccharide, meaning instead of just two sugars linked together, it's long, long, long chains of the same sugar on repeat. Plants, algae, and plant-like protists have cell walls made out of cellulose, while fungi have cell walls made out of chitin. So whether it's a fungal cell wall made out of chitin or a plant cell wall made out of cellulose, they have the same feature of preventing overfilling with water. Okay. Now, I'm also noting in the slide that insects have chitin, but it's not around each cell. So it's not a cell wall. For insects, it's not a cell wall made out of chitin, but rather their exoskeleton. So you know how insects have their skeleton on the outside instead of on the inside? So that hard outer shell on insects is their exoskeleton, and that's made out of chitin as well, which is just sort of interesting because insects and fungi are the only two places that you find chitin in nature. So here's the chitin cell wall for fungi. Some protists have a cell wall, and some have a glycocalyx layer. Those with cell walls are typically more plant-like, while those with glycocalyx are typically more animal-like, but that's not a strict rule.